Now we move on to the next set of organisms, and the quickly discussion of HBB is going to happen. What is HBB? Haemophilus, Bordetella, Brucella. Let's take three slides and finish it off. When I talk about Haemophilus, the first Haemophilus that I am going to talk about is Haemophilus influenza, which is also known as the fever bacillus. It is also known as Pfeiffer's bacillus. कितना ज़्यादा F आ रहा है मैम? That is going to be Haemophilus influenza fever bacillus. Yes, this is going to require some FF factors which I'll tell you. But please note, this is having a capsule. If it has a capsule, this means it has 6 types. Typing हमेशा होता है. And please note, the most important one is HIB, because HIB is the one, B type is from which you make a vaccine. So vaccine के लिए and virulent वाला is going to be the HIB based on a capsule. What all does it cause? We all know Haemophilus influenza बच्चों में meningitis कराता है, and meningitis तो ठीक है, but influenza है तो obviously your respiratory tract involvement has to be there. Laryngotracheobronchitis, pneumonia, and other suppurative lesions of the body are seen. I'll finish off the treatment quickly, which you know is done by ceftriaxone usually for Haemophilus influenza. But what did I say in the beginning? It is a Haemophilus influenza fever bacillus. What all factors does it need? It needs factor 5 and factor 10. Actually, honestly telling you, these are V and X. These are V and X, but most people call it 5 and 10. We will also call it 5 and 10. So you know who is going to provide this factor 5 and 10. So remember, Haemophilus influenza fever bacillus needs factors. So this requires factor 5 and factor 10, which is going to be given by the blood agar. So ma'am, they are not actually hiding in the blood. You will have to break the blood, then they will come out. So how will I break the blood? I take a blood agar over here and I put a Staph aureus. Center में क्या करूँगी? I'm trying to act smart. I put a Staph aureus. If I go back to the first slide of my rapid revision, I have studied that Staph aureus breaks blood. तो इतना area में will Staph aureus break the blood? Does it cause hemolysis? Yes. If it will cause hemolysis, then I will say, oh, blood has broken, factors released. मतलब party time for Haemophilus. Haemophilus will say, wow, यहाँ पे factors हैं, let me go and grow over here. Can you say Haemophilus influenza has come and has started growing over here? Haemophilus influenza has come and started growing over here. So satellite हो गया ना. If this, for example, is going to be a Staph aureus, Haemophilus is saying like a satellite, I'm going to grow around it, because Staph aureus तो party करा रहा है, treat दे रहा है. Staph aureus is releasing all the factors. तो यहाँ पे party है, let me go and roam around it. So Haemophilus is satelliting around it. So that is referred to as satellitism. Very important question. There's another Haemophilus which is important, which is called Haemophilus ducreyi. So सबसे पहले from ducreyi you will learn croid. Ducreyi causes ducroid, that is chancroid and chancroid. इतना cry sound आया ma'am. When you said ducreyi, I was crying. When you said chancroid, I was crying, and crying with pain, because these are painful genital ulcers. Painful genital ulcers. So remember, it is a soft chancroid. Painful, and who are the people who cry? They are said that, you know, they are number one, they are crying because they have a lot of pain. So crying because यहाँ pain है. And they are very soft-hearted people. They are nice people. They express themselves. That is why they are crying. So soft-hearted अच्छे लोग हैं. They are just crying because they are in a lot of pain. So please note, when they say Haemophilus ducreyi, you will say croid, and you will say, oh, soft-hearted person is crying with pain. Soft-hearted person is crying with pain. These are painful ulcers, because I hope you remember in syphilis, I am writing it syphilis. Why? Because everything is painless. ये बिल्कुल hard, made of steel, rock, stone, very, you know, not a soft person. You're gonna see a hard, painless chancre that you see over here. Syphilis में सब कुछ painless है. He is not crying. He doesn't have any pain because he's made of steel. He's hard. ठीक है? That is how you learn. So ducreyi is going to be crying with pain, and that is going to be a soft ulcer. What else? Now you say that acha, he's crying so much, ma'am, he's in so much of pain. So what do? Someone's not in a good mood, thoda sa chocolate de do. You give him some chocolate, you give him some nutrition, give him some food, he will be happy. So what are you going to do with ducreyi? You're going to grow it in a chocolate agar. Regular chocolate agar? No, you're going to give him extra chocolates because he's a little sad right now. So you supplement it with little growth factors like Isovitalates or serum, basically multivitamins ki tarah, Revital ki tarah. Kuch extra you are giving him because he's crying. Poor thing. Okay, so remember this crying kid is given chocolates, and along with chocolates, little vitamins like isobutalics and fetal calf serum. Have I already told you that hemophilus ducrey? I'll remind you of that mnemonic 1 millionth time more. That D banaya tha yahan se, and hemophilus ducrey also shows you the safety pin appearance. Yes. And what is the treatment? Azithromycin. So ultimately you realize there are so many hemophilus organisms. Some of them require factor 10, some require factor 5, some need CO2, some are going to show hemolysis. Kaise yaad rakhenge? Jaldi and very simple. My favorite table, because I can finish it in 30 seconds. Which are the ones which will show you hemolysis? Ma'am, jinke naam mein hemolysis hai. Hemolyticus, parahemolyticus means these are the only two which show you hemolysis. Baki I'm not writing negative. ठीक है, which are the ones which need CO2? CO2 मतलब afro word means CO2. So if I pick up Afrophilus and Paraphrophilus, these are the only two which show me CO2. बाकी सब में वही negative. बड़ा simple है ये तो, right? Now coming to the next, which are the ones that only need factor 5? Ma'am, 5 मतलब हम 5 penta, 5 और penta. Ma'am, all the P वाला मतलब para wala organisms will need only 5. What are the para? Dhundo para influenza only 5, not 10. Then parahemolyticus only 5, not 10. Paraphrophilus only 5, not 10. 5. Which is the one that only requires 10? Which is the one that only requires 10 or 10? So that is Ducrey. Ducrey requires only 10. So that is going to be 10. What is 10? 10 is going to be for Ducrey. 10 is going to be only for Ducrey. हो गया. तो 5 वाला is all para. 10 वाला is Ducrey, and whatever is left, dono. Who will need dono? So ma'am, bhai. When will the bhai come together? What is this bhai mnemonic? So bhai means both are needed for hemolyticus, aegypticus, and influenza. For bhai, when I say bhai, you will say, ma'am, both factor 5 and factor 10 are needed for hemolyticus, okay, then for aegypticus, and then finally for influenza. And the table is done. I told you na 1 minute ka table hai, to simple. Hemolysis wale mein hemolysis dalna, CO2 wale mein put the afro things, and then bhai mein you will put hemolyticus, aegypticus, and influenza. Only 5 mein you will put all the para people over here, 5, and only 10 mein 10, that is Ducrey. Hemophilus is also done for you. What is the next one that I'm going to start off with? Let's see if you could identify this very famous disease. What did I say? Whooping cough or pertussis. This means now I'm going to start off with Bordetella pertussis. Very good. Bordetella pertussis. No points for guessing. Causes the pertussis or the whooping cough, also known as the 100-day cough. Am I going to tell you the toxin? Never, because pertussis toxin you know. I've given you a mnemonic called cyclic AMP, and P was for pertussis, so this increases. Cyclic AMP. So what are the phases in which someone has this whooping cough? So starting wala to catarrhal phase hai. Ma'am, thoda fever, thoda cough, nothing worrying about. But although it sounds like very catarrhal, thoda, this is the one that is going to show you maximum infectivity. Maximum infectivity is going to be seen in the catarrhal phase. Very famous question. Then comes the real deal, the paroxysm. This is the time when the person has the whoop, that barking cough, right? He takes a big air ka gasp inside. That whoop is seen over here. And finally comes the stage of recovery. Convalescence always means recovery. So remember, initially there's a very catarrhal, simple wala, chota cough, chota fever phase, but that is very infective. Then comes the real whoop wala, whooping cough phase, and then comes the recovery. So what is the drug of choice? We can give erythromycin or azithromycin. And is there a vaccine? Yes, we use an acellular vaccine. Earlier they used to use a cellular vaccine, but that ended up causing encephalitis in patients. So now they don't use cellular vaccine. Now the problem is that you only have to use an acellular vaccine. You cannot. DPT mein jo pertussis hota hai na, that is acellular, and that has these 4 things. You don't have to learn their full form. But PT, you know, pertussis toxin, filamentous hemagglutinin, something for aggregation, something for pertussins ka pert. So aise hi rat lo is what I would say. Don't learn the full form. Anyway, pertussis toxins and pert, so you will anyway learn na. You just need to learn FHA and AGG. Something with agglutination, something with hemagglutination. So ye dono yaad rakh lo. These are the 4 things. This is the tetrad that a DPT vaccine ka pertussis has. Acellular, never a cellular vaccine. But guys, I didn't tell you the culture media. Because that is so simple, na? And when I'm talking about Bordetella, common sense, we are gonna use the Bordet Gengu media. And BBB, Bordetella, mein Bordet Gengu shows you the bisected pearls appearance. They look like nice pearls over here. And they show you the bisected pearls appearance. But imagine if these bisected pearls end up fusing, sab aapas mein fuse kar gaye, what is that gonna show you? Now that is gonna become if it becomes confluent, it is going to be shown as an aluminium paint appearance. So pehle to they are pearl, then they fuse and form aluminium paint appearance. If you look at this under the microscope, it's very difficult to visualize, but these are looking like prints. I'm trying to make a thumbprint appearance over here. So Bordetella is actually going to show you the thumbprint appearance under the microscope. So bas Bordetella story is done. Cyclic AMP is the toxin. Which kind of vaccine do you use? Acellular vaccine. Which is the most infective stage? Most infective stage is going to be the starting catarrhal phase. What is the culture media? Bordet Gengu media for Bordetella. Next, you're going to have bisected pearls appearance, and then you have an aluminium paint अगर वो fuse कर जाए. ठीक है, microscopically it is a thumbprint appearance. Also, if you remember from your main videos, I told you, does Bordetella have a capsule? Yes, I told you na, Bordetella की body है, body बनी हुई है, मतलब this is gonna be a capsulated organism. If you want to recall that, you definitely can. So Haemophilus done, Bordetella done. What are we left with? We are left with Brucella, which we know it is a zoonotic disease. Cows and animals and dogs and pigs in सब में होता है. तो cow बोला ना, cow मतलब it is cow positive. Brucella, which is a cow positive, मतलब catalase, oxidase, urease, सब कुछ positive. Cow should remind you of cow, animal वाला cow, and Brucella is definitely to do with cow, right? So catalase, oxidase, urease positive. There is a triad of Brucella which is important. These patients come to you with night sweats, arthritis, and HSM, hepatosplenomegaly. Also in pathology, please note that sometimes the platelet count can also be decreased. Thrombocytopenia can be seen. That is Brucella. So, kaise diagnosis? Ma'am, hang on. This is zoonotic disease. Animals may be hai, humans may be hai. So, diagnosis you will have to make in both places. In animals also, in humans also. So, animals ke liye to I will do a milk ring test. If I see a purple color ring on the top, I am saying that, oh, okay, animal has antibodies. Matlab, this is the milk ring test which is going to be done in animals. And naam se pata lag raha hai na, milk. Milk will of course come from animals. And ring, if I see a purple color ring in the top, this means there is going to be a positive milk ring test that I've got. Whereas what is done in humans? In humans, we do the standard agglutination test. What is standard agglutination test going to tell me? It's going to tell me IgG and IgM antibodies are going to be detected. In humans, I will be able to detect IgG and IgM antibodies. However, if you say, nahi, I don't want to detect IgM. I am not interested in IgM. IgM ko maar do. Remove the IgM. How will I remove them? IgM मारना है तो मारने के लिए mercaptoethanol. If I treat the blood sample with mercaptoethanol, IgM is destroyed. So remember how I have learned this. Mercaptoethanol मतलब IgM को मारना, right? IgM को मारना is mercaptoethanol से use करना. This is a question that you get in a standard agglutination test for Brucella. If you treat with 2 mercaptoethanol or 2Me, which is the antibody that is destroyed, तो क्या मरेगा? Mercapto से क्या मरेगा? IgM मरेगा. So they will ask you, then what are you going to test? You will be testing IgG. Very, very famous previous year question. Finally, what is the treatment? I think we all know. There's doxycycline and for 45 days and streptomycin IM, which is given for 14 days. There are certain alternatives which have been given by WHO, which I'm sure you guys have read about. I've taught you in the main videos. But yeah, that's the main treatment that you have. So the HBB group is also over. Haemophilus, Bordetella and Brucella. Having discussed the gram-positive cocci, negative cocci, positive bacilli and negative bacilli, now we come down to ones which do not stain well with gram. And those are MRCS, that is Mycoplasma, Rickettsia, Chlamydia and Spirochetes. And let's start with the opposite order. That is, first we're starting with the family of Spirochetes. And what all comes under it is Treponema, Borrelia and Leptospira. So if you look at Treponema, T is going to have very, very tight coils. You can see that it's a spiral. All of them are spiral, but it's very, very tight, looking exactly like a screw, right? And that is why the motility is said to be the corkscrew motility. Next, coming to Borrelia. Borrelia, as the word says, Borrelia has very, very loose spirals. They are not very tight. They are loose coils, and lula, that is how we've learnt it. It is going to show you the lashing motility. So remember loose spirals and lashing motility. Finally comes to leptospira. Leptospira is lapeta hua. That is how we've learnt it. Leptospira means it's going to have hooked ends. And is this also going to show you motility? Yes, it is. This is also going to show you a motility. No special name given to it. So please note that in all of these, I did not show you any flagella coming out. If there's no flagella coming out, how are all of these moving? How are they motile? Because remember, spirochetes don't have a flagella coming out. They have a flagella inside. All of them have an endoflagella. Which part of the bacteria has an endoflagella? That is the periplasmic space. Let's begin with the first one. What am I starting with? The tight wala coils. Tight wala coils matlab I'm starting with treponema, and the treponema that we are going to study is treponema pallidum, which causes sexually transmitted disease called venereal syphilis. The incubation period, which you will always remember, 9 to 90 days, is how syphilis is going to occur. And how many stages does it occur in? Primary syphilis, secondary and tertiary. So I've got you the 3 stages put up all in one go. So what is this? Primary, secondary as well as tertiary syphilis. So remember, from today onwards, instead of syphilis, you will all learn it as syphilis, because if they ask you what is the status of the ulcer and the lymph node, everything is painless. The genital ulcer is painless and the lymph node is also going to be painless. What is that known as? That is known as a hard chancre. That is known as a hard chancre. I hope you remember this has to be differentiated. Right before this, I taught you something called chancroid. Chancroid was crying with pain, and soft-hearted people cry with a lot of pain. So that was a soft. This is painless, bilkul hard. This is painless, no crying happening over here, and this is a hard, stone-hearted person. So hard chancre, which is painless. Ulcer is also painless. Lymph node is also painless. So I called it painless. Syphilis. Now we come down to secondary syphilis. Everything should be reminded of 2 in this. So firstly, you can see that there are a lot of warts that I'm seeing at the mucocutaneous junction. These are referred to as condylomata lata. I've always learnt it as condylomatu latu, because that will remind me of obviously the situation of these secondary syphilis. So condylomatu latu is how you're going to learn it. Next we have rash. So your 2 hands and 2 feet are going to show rash. So again, something which you see in 2 hands and 2 feet. So that is the secondary syphilis. Now coming to the tertiary syphilis, which shows you the very famous gamma. You can see a gamma. I hope you remember from pathology that gamma is going to show you the presence of granuloma. And other than that, it shows you cardiac and CNS involvement. CNS may it causes general paresis of insane. I hope you know from medicine now that general paresis of insane, may say the insane word has been removed and now it is just known as general paresis. And in cardiovascular, please note that there is aorta involvement. In fact, in pathology, you study a very famous thing known as the tree bark aorta. We study the tree bark aorta, and also this can cause aortic regurgitation. So the aorta is basically affected in syphilis. So primary is going to show you the ulcer, painless syphilis. Secondary, condylomatu latu, and there are going to on what junction? If you want to complete it, condylomatu latu on the muco-Q2 junction. So mucocutaneous junction, and 2 hands, 2 feet showing you rash. Tertiary is going to show you all organ involvements with CVS, CNS, or neurosyphilis, cardiosyphilis, and it is also going to show you gamma formations. How do you do the lab diagnosis? We know that spirochetes are very patla. You can't see them on a light microscope. So you're going to use something called a dark field microscope, and you can see that spiral looking tight coils wala spiral looking organism out here. But if you say, I don't have a dark field microscope in my lab, I have a light microscope. Now what do I do? Patle ko mota karo. If you have a very thin organism, cover it with lots of clothes. I'll cover it with lot of clothes and it's going to become a mota organism. So cover it with what? Silver impregnation. There is something known as the Levadity stain, and we have also studied the other one, and that is known as the Fontana stain. These are the 2 silver stains that you have. So please note F for F. Fontana stain is basically used for the fluid sample, and Levadity, when I write Levadity, the TI is going to remind you of the tissue sample. So if they say there is an ulcer ka biopsy or a tissue given, you will say, oh, tissue hai to Levadity wala silver I will use. If they say there is a film that has been prepared solid, a film or a fluid or a discharge, कुछ भी film, fluid, discharge ऐसा है तो I will use the Fontana. So F for F and Livardi T for tissue. This is how you do the biopsy wala diagnosis. How about the antibody diagnosis, the serological diagnosis? There are some non-specific and some specific tests. Under non-specific, the first one is Wassermann test. You studied this in your immunology. It is a complement fixation test, but no one does it now. The other one is a Cahen test, which is a tube agglutination test. Again, not done now. The two that I have to know under non-specific are VDRL and RPR. These are definitely done nowadays. And you need to know the difference between these two. Then I'll come to the other variety. So what is the difference between VDRL? You all remember the full form now? Venereal Disease Research Laboratory. So VDRL and RPR ka many students I have seen, they do not know the full form. Rapid Plasma Reagent. Right? Rapid plasma reagent. So remember Venereal Disease Research Laboratory versus RPR, what is used, how is used, and what is better or less. Let us start. So this VDRL is, how I have learned it is very, very difficult reaction in the lab. Very difficult reaction to do in the lab is VDRL because every step is problematic. You will say that, ma'am, if you take blood sample from patient, you want to see his antibodies for this spirochetes. If you take the blood sample, you will have to heat it. Pehla problem. You can't use it directly. There is a preheating that is required. RPR says, ठीक है, हटा दो. I will not have to preheat, direct use करो. RPR is trying to be very rapid. RPR will try that it overcomes all the problems. VDRL is very, very difficult test. RPR says, I will make your problems easy. So VDRL says, pehle to you will have to heat the sample. Then after that, the antigen that you will add, that antigen is a cardiolipin antigen, and once you have prepared it, you will have to use it. You will have to use it within 24 hours. So you will have to prepare an antigen. कौन सा antigen? Cardiolipin. क्या होता है? What is cardiolipin antigen? Cardiolipin antigen is cardio, ma'am. Someone's heart it is going to come out of. It is going to come from the beef heart. It is going to come from the beef heart, and lipin matlab it has all lipid things. It is lipid coming out from the beef heart or lecithin or एक और lipid जैसे cholesterol. तो cardio and lipin मतलब all lipid thing, and cardio means beef heart. So you will have to require cardiolipin, and once you prepare it, you will have to use it in 24 hours. So antibody के लिए भी heating करो, antigen के लिए भी you use cardiolipin, and then when you finally make them react, you will have to look at it under a microscope. So this is a slide based test. Then again, you need a microbiologist and a pathologist to see it under the microscope. हर जगह problem है मैम. Whereas RPR says that you do not need any preheating. You don't need this. You don't have to prepare anything that can be used within 24 hours. No fluid is needed, and you don't even need any microscope for this. ये card पे test होता है, तभी तो it is rapid. Have a look at this picture. This is a simple card-based test, so you will just have to read it with your eyes on a card. You don't need a microscope. All your problem is solved. So obviously the RPR is what is very, very rapid. But please remember, in VDRL, all samples can be used. For example, you can use a blood sample, you can use a CSF sample, whereas RPR says अब जल्दी में सब कुछ नहीं मिलेगा. Rapid revision, if it is rapid revision, we're not doing 50 hours of syllabus, we're doing 15 hours of syllabus. So सब कुछ तो नहीं मिलेगा, important मिलेगा. So RPR says there is no CSF testing that can be done, which gets me to a very, very important question. What is the investigation of choice for neurosyphilis? Because in neurosyphilis, I will be getting a CSF sample. So ma'am, CSF ke liye, if you want to get like the top rank, you will have to listen to the main videos. Only RR is not enough. So please note, VDRL can even test the CSF sample. So for neurosyphilis, you will always do VDRL. Very important question. So the non-specific tests are done. Non-specific matlab they will come in many other diseases also. So I will have to opt for a list of specific tests which students find very difficult to learn. So let me simplify all these specific 4 tests for all of you. Then I'll tell you sensitive, specific and all of that. So remember, FTA, you don't have to know the exact full form, but yeah, thoda aaspas ka you should have an idea. Fluorescence treponemal antibody, ABS, is going to stand for absorption assay. So who are the people who absorb everything? Ma'am, sensitive people, happiness, sadness, emotions, they absorb everything. So which is the most sensitive test? The absorption wala. FTA absorption is the most sensitive. Next is treponema pallidum immobilization. Immobilization matlab very rigid. Matlab if it is here, it is only here and not going anywhere else. Means it is specific. If it is coming for treponema, it is only coming for treponema. So that is going to be the most specific. So absorption wala is sensitive and immobilization wala is specific. However, the problem is that treponema pallidum immobilization assay uses live treponemes. I don't want to use live treponemes in my lab and have a problem for everyone around me. So what am I going to do? Actually, then I take help of TPPA, treponema pallidum particulate agglutination test. So if they ask you in terms of specificity, theoretically speaking, TPI is the most specific, but uske baad actually practically TPPA is what is done. Finally, we have treponema pallidum heme agglutination assay, which is TPHA. So asking you if these are the 4 options given to you, what will you say? You will say FT absorption and absorbing sensitive people, treponema pallidum immobilization most specific. Second most specific is treponema pallidum particle agglutination, and finally we have heme agglutination. Okay, and if I ask you what is the investigation of choice for neurosyphilis out of these, you know for neurosyphilis the very difficult test has to be done, and that is BDRL. Okay, now let's move forward. You are obviously wanting to know the treatment. That is going to be benzathine penicillin. So tight coils wala treponema is done. Thoda loosen out karte hain. Loose coils wala Borrelia. So now Borrelia is of 3 types. Borrelia burgdorferi, Borrelia recurrentis, and other ones that I'm going to tell you. 2-3 Vincenti, these are going to come. So burgdorferi causes what? This is one of the most common vector-borne infections in US. The history will always start as a resident of United States of America, and that disease is known as the Lyme's disease because it is the bite of the what? It is the bite of the heart tick, that is the ixodid ticks. Ixodid ticks or heart ticks are going to basically go and bite, and jahan wo bite karenge, can you see this kind of a rash? There is a redness, then there's another aisa concentric bullseye kind of lesion ban raha hai, ma'am. There is something called a bullseye lesion that is forming. So you can call it a bullseye, you can call it a target, you can call it a ring or an annular, whatever you want to call it, but actually it is erythema chronicum migrans. Erythema tells me bilkul it is a red color. Migrans tells me that it is gonna move. As you can see, it's going outside, and as it is going outside, the color in the center will become lighter. So it keeps spreading outside. Erythema chronicum migrans. This is the same picture you will get, and they will want to ask you always and always about Lyme's disease. That is Borrelia burgdorferi. Now coming to the other one, that is Borrelia relapsing fever wala, ma'am. There are 2 types of relapsing fevers, the one that is caused by louse and the one that is caused by tick. So which is caused by recurrentis and which is caused by Dattoni. So Dattoni mein T aata hai, so that is going to be the tick-borne relapsing fever, and recurrentis is going to be louse-borne. How have I learnt it? So dekho, louse sounds like love, and this is a very famous first aid ka USMLE mnemonic. Love is going to spread like an epidemic between whom? It is going to spread between human to human, and love should be obviously something that should happen again and again. You should again and again fall in love with the same person. Okay, so having said that, that love for louse, it is going to spread like an epidemic. So it is the epidemic relapsing fever again and again with the same person, that is Borrelia recurrentis, and human to human is how it is going to occur. Whereas tick-borne mein Dattoni aayega, and that is going to be endemic. What is the treatment for this? Because again, relapsing fever ka they will say relapsing matlab fever aayega jayega. Relapses of multiple 4 to 10 relapses of fever can come. Patient will have fever, then few days he's fine, then he'll again have fever, and few days he'll fine. So the treatment is going to be tetracycline or chloramphenicol. One more thing, please collect the blood sample during the fever, during the pyrexia, because when the fever will be present, the organism will be present. When the fever will be absent, the organism will be absent. That is how it works. So take a blood sample while the patient is febrile. Last one is Borrelia vincenti, which obviously causes something in the mouth known as Vincent's angina, and that is an ulcerative gingivostomatitis. So as I said, Vincent's angina happening in the mouth. Borrelia ho gaya, loose spirals. Ab lapet do. What is that lapeto thing known as? Lapet do means the ones that is going to have hooked ends. The hooked ends ones is going to be leptospira. What does it call? Lapet do. Wheel bana do. Okay, so this was for example kind of a spiral. अब क्या करोगे? उसे ऐसा लपेट दिया, wheel बना दिया. What is Weil's disease? Weil's disease, ma'am, is icterohemorrhagic fever. Let's tell you the symptoms. Ictero, patient is going to have jaundice. Hemorrhagic, patient is going to have hemorrhage, also known as hepatorenal syndrome. There is involvement of liver and there is involvement of kidney. Wow, ऐसा होना चाहिए. The name should tell me everything. Ictero plus hemorrhagic plus hepato plus renal, that is going to be the Weil's disease. And this has the very famous, so from today onwards, you will call it leptospira. That is how you have to learn it, because this is gonna have the rat urine, and the rat urine will then contaminate what? It will contaminate the rainy water. It will contaminate the rainy water, and who will walk on that rainy water? The rainy water will go to the rice water fields. The rice fields. So the rice field workers will basically get infected with it. So remember, whenever they say rat urine has contaminated rainy water and that has been affecting the rice water field workers, you know it is a case of leptospira. So ये आखिरी R क्या है? Because this tends to show you the Dinger's ring. And Dinger's ring is what you can see over here. Can you see there's a ring? दो है. There are two test tubes. In one I don't see a surface है, दोनों का surface. In one I don't see a ring, and in the other I see a ring. What medium is this? EMJH media. This tends to grow on the EMJH media, and that is going to be the Dinger's ring. So that is how you do leptospira. But best diagnosis तो है मैम antibody निकाल लो. How do I find out the antibody? By the microscopic agglutination test. So what all agglutinations have I studied? Where did we have standard agglutination test? Standard agglutination test I studied a while ago. You studied it in Brucella. Microscopic agglutination test you are studying for leptospira. I'm not giving you a mnemonic right now. I'll give you a mnemonic in 5 minutes, because there's something called cold agglutination test in mycoplasma. बस वो पढ़ के then I'll give you a beautiful mnemonic which came in the NEET 2023 also. So now please come back to the next thing, and that is done with this spirochetes family. So we were doing MRCS mnemonic, na, the ones which don't take any gram stain. So spirochetes is done. Now let's move on to the next R and C, that is going to be chlamydia and rickettsia. So what is chlamydia, guys? Chlamydia is an ABC वाला organism. ABC मतलब Firstly, it is A, मतलब it is an atypical. बिल्कुल atypical तो है मैम. It is not taking up the gram stain nor gram positive negative, कुछ भी नहीं है. One more thing that makes it atypical is that it is not going to grow on any of the culture media. It cannot be cultured. It is an intracellular growing organism. So remember, A for atypical bacteria, C for it cannot be cultured. And do you know what else makes it atypical? Because it doesn't have the peptidoglycan cell wall. It doesn't have a peptidoglycan wala cell wall. That is what another thing that makes it atypical. Also, it is quite a lalchi bacteria, greedy bacteria. It's going to take ATP from the host. So these are actually said to be ATP parasites. Don't make them parasites. Parasites only because it's trying to take out ATP from me. So A or C to ho gaya. These are atypical. Why atypical? Because they don't have a peptidoglycan cell wall and they cannot be cultured. Another thing, they are ATP hosts, or they are ATP parasites that you have. And finally, B for basophilic. They are going to show you basophilic inclusions, and that's the B that you have. So chlamydia is ABC. Okay, now we ask you, what are the two forms in which chlamydia is going to be present? So you will say, ma'am, एक तो है जो हमें infection कराएगा, the one that is going to enter into us, and the one that is going to replicate into us. So please remember, this is something called an elementary body, and the other one is going to be known as a replicative body, and or in other words, you know, it's some books also call it a reticulate body. ठीक है, elementary body and reticulate body. And now I can easily understand R for R. Reticulate body is the one that is going to replicate in our body. This is the replication wala. And remember, R for R, it has RNA more than DNA. On the other hand, if I say elementary body, you will say, ma'am, elementary body EE lag raha hai. The one that will enter us and exit us. The one that is going to enter us and exit us, that is going to be the elementary body. And E for equal, the DNA and the RNA is going to be exactly equal. How easy to learn this mnemonic also. So repeating, there is an elementary body which will enter and exit. मतलब they ask you infection कौन कराएगा और जाएगा. Enter, exit is elementary, and equal, DNA, RNA equal. Reticulate body is the one that will replicate, make many copies in us, and that is going to have more of R, that is more of RNA. Done. Now how many types of chlamydia do we have? Chlamydia trachomatis, A, B, B, A, C. This so you all know. Chlamydia trachomatis A, B, B, A, C causes trachoma, the most common infectious cause of blindness in our country, right? Whereas C के आगे चलो, D, E, F, G, H, I, K. These are the ones which cause very commonly non-gonococcal urethritis. Non-gonococcal urethritis मतलब discharge होगा. Discharge matlab that discharge can also be, the discharge can come in contact from the urethra, it can come in contact with the fingers and you can put it into your eyes. That is known as inclusion conjunctivitis. That is known as inclusion conjunctivitis. Now through that canal, if the baby passes, the baby is also going to have a problem. So these can also land up with ophthalmia neonatorum. These can also have ophthalmia neonatorum. So please note that none of these cause blindness. Neither inclusion conjunctivitis, right? Neither inclusion conjunctivitis is going to cause blindness, nor this ophthalmia neonatorum. Maybe ophthalmia, yes, but inclusion conjunctivitis does not. So remember that trachoma, ABBAC causes trachoma only, but D se aage jo hai, they first cause an NGU, non-gonococcal urethritis, from there inclusion conjunctivitis, and in children, neonates, ophthalmia neonatorum can happen. Now we come to the serotypes L1, L2, L3. Common sense, L1, L2, L3 will cause L for lymphogranuloma venereum. These were all the trachomas that you had to know. And now I have to tell you what is lymphogranuloma venereum. You remember there was a mnemonic called ABCDEF. So jaldi se kar lete hain. What is that? Usually these patients are asymptomatic, but when they have an infection, they are going to show you these very painful lymph nodes. That is buboes. So please remember, lymphogranuloma venereum is what? You will say, ma'am, वैसे तो ulcer होगा अगर genital पे तो painless है, but because lymphogranuloma venereum has L in it, so the lymph node tends out to be painful. Then that painful lymph node is referred to as the buboes. Now we come to what is it going to show you? Which organism? Chlamydia trachomatis. कौन सा वाला? L1, L2, L3. Also, it can show you something called cup. What is this syndrome? Conjunctivitis, urethritis, and polyarthritis. This is known as the Reiter's syndrome. Finally, talking about the complications, strictures can occur. That is known as esthesiomenes. So, genitals में strictures can occur. If you have to do a skin test, what is the skin test? It is known as a Fry's test. Remember, Fry's test is a skin test for LGV. And is there any complication? Yes, Fitzhugh-Curtis syndrome. What is Fitzhugh-Curtis syndrome? It is a perihepatitis. It is a liver involvement. Where else did I teach you Fitzhugh-Curtis syndrome? If you remember, over there also I told you I will teach you with Chlamydia trachomatis. The other place was Neisseria gonorrhoeae. That also shows you the Fitzhugh-Curtis syndrome. Finally, with G, it shows you a groove sign, which is so beautifully seen over here. You will appreciate that there are lymph nodes on one side over here and a lymph node on one side over here, and there is a gaddha in the center. Groove stands for gaddha. So that groove is basically made by the inguinal ligament. So the lymph nodes are separated by the inguinal ligament. So there's a gaddha of inguinal ligament and lymph nodes are on one side, and on the other side, that is a groove. The treatment bhi bata do. Treatment is going to be drug of choice, doxycycline. So remember, buboes are seen which are painful. Chlamydia trachomatis, cup syndrome or Reiter's can occur. Drug is doxycycline. Complications में esthesiomenes, and another complication is Fitzhugh-Curtis syndrome. Skin test is Fry's test. Most important, groove sign will be seen. That is lymphogranuloma venereum. So yes, coming back, your chlamydia point is totally done in terms of trachomatis. But we also have something causing pneumonia, chlamydia pneumoniae. Always remember pneumonia as pneumono, because how many strains does it have? It has a single strain, and that single strain was from Taiwan. So that is known as the Tuor strain. It caused pneumonia in Taiwan. So that is Tuor. Pneumono ke paas single strain hai, whereas Sitiki has several strains. It has several serotypes which are available, and that is what is chlamydia Sitiki. So that is going to take care of all of your things. But I want to ask you a question from ophthalmology. Are there any bodies that you see in trachoma in ophthalmology that they ask you? Have you heard of Halber-Steeder-Prowaski bodies? Yes. Halber-Steeder-Prowaski bodies are going to, so most of the places you don't see the full form only. They write it as HP bodies, right? Halber-Steeder-Prowaski bodies are seen in trachoma, which is very, very important information that you have. Whereas there is another question that you end up seeing, that is, where do you see the Leventhal Cole Lilly bodies? Where do we see the Leventhal Cole Lilly bodies? LCL it will be written as, and that is seen in chlamydia Sitiki. These two bodies are important. So repeating, Halbersteder-Prowaski bodies. So Halbersteder-Prowaski bodies are seen in trachoma and LCL bodies are seen in Sitiki. So all of your chlamydia is done. We can move on to the next deadly family, and that is Rickettsia. Before I start with it, I hope all of you remember that Rickettsia is something which is going to be arthropod borne. So there is something that is going to be involved over there with a tick, mite, louse. Some arthropod is going to be there, and whenever an arthropod will bite, this means the rash will be there. So all of them are going to show you the presence of a rash. There are exceptions which I'll tell you in the end. So first, these are all the different Rickettsia typhus group that you have to know. You need to know their organism and you need to know whichever arthropod is involved. So firstly, now you need to get familiar with one column. Okay, so when I say epidemic has P in it. So the organism is going to be Rickettsia-Prowaski because that also has a P in it. Then when I say अच्छा endemic में तो अपना वाला नाम रखो ना, endemic is अपना वाला, so that is Rickettsia typhi अपना वाला. So typhus is gonna have typhi and Prowaski is gonna be epidemic. ठीक है. Now we have Rocky Mountain Spotted Fever, R for R. Rocky Mountain Spotted Fever is gonna be Rickettsia rickettsii, again R for R. African tick typhus, no points for guessing. Rickettsia africae. So Rickettsia africae is the one that is going to show you the African tick typhus. Indian tick typhus, how I have learned it is, India में कोरोना बहुत हुआ था, तो कोरोना का common थोड़ा change करो, coronary. Indian tick typhus is going to show you, is going to be caused by Rickettsia coronary. Now we come to rickettsial pox, which is said to be the mildest of them all. Remember, rickettsial pox is the mildest infection of them all, and that is caused by Rickettsia acari. And finally, the scrub typhus, S for S, that is caused by Orientia sussugamushi. So remember, scrub typhus by sussugamushi. ठीक है, चलो हो गया. Epidemic for Prowaski, endemic अपना वाला नाम typhi, Rocky Mountain Spotted Fever for Rickettsia rickettsii, India में कोरोना तो coronary, Africa of course Africae, rickettsial pox by acari and scrub typhus by sussugamushi. Are all of them going to have an arthropod? Yes, I'll be telling you. Will all of them have a rash? Yes. So please note, in all of them, the rash is going to first begin on the trunk and then it is going to go on the extremities, right? So it's going to go from the trunk and then it will go on to the extremities. However, in Rocky Mountain Spotted Fever, ulta hai. It first starts on the extremities and from there it is going to go on to the trunk. Basically, the concept is, how I have learned it is, Rocky Mountain, mountaineering kare, mountain climbing kare, what do you do? Which part of your body comes in contact with the mountains? Your hands and your feet. So extremities पे exposure है, तो extremities में rash occurs first and then it goes on to the rest of your body. In all the others, it's the opposite. Now let's talk about the vectors or the arthropods, and for that I have a mnemonic called Let-Fentria PSM. What is let? So firstly, L will tell me that I'm dealing with the louse. So now tell me L for louse and the other one over here is going to be F for flea. One of them is epidemic typhus, one of them is endemic typhus. So fen में N आ रहा है. This must be the endemic typhus. This automatically means that the louse is going to be epidemic typhus. Right? Now we come to tria. Ma'am, tria mein to this T is sounding like a tick, and now I know the ria is going to be Rocky Mountain Spotted Fever, Indian tick typhus and African tick typhus. So unme to tick word is only coming. Finally, I come to PSM. M stands for mite, and now I know P and S is going to be number 1 for rickettsial pox and S is going to be scrub typhus. Dobara kare, Let-Fentria PSM. So first write down. When I say let and when I say fen, so flea is going to be for endemic typhus and louse is going to be for epidemic typhus. Then I'm going to write the mnemonic tria. What is tria going to be? So tria will remind me of Rocky. Tick is going to be for these three: Rocky Mountain Spotted Fever, Indian tick typhus, African tick typhus. And finally I write PSM. In PSM, mite is going to be for these two, that is rickettsial pox as well as the scrub typhus. Ho gaya table yaad? This is what you were very scared of, but now I'm going to twist the table. Now tell me, this epidemic typhus happened because of what? Prowaski, because of which arthropod? Louse. This happened in a person and after some time it slept off, latent ho gaya. And after some time it woke up again, got reactivated. What is that typhus known as? Where something is going to sleep off for a while and then going to wake up later. That is a reactivation typhus, recrudescent or Brill's ulcer disease. This is just a variation of epidemic typhus. So is this a new typhus that has occurred, ma'am? Will I have to learn another vector, another lice, flea, mite, something? No. This is just a sleeping and a waking up reactivation scenario. This is not a new vector that you have to know, and this is going to be less severe. That is Brill's insert disease. It's a variant of epidemic typhus. Sleeping, waking up story is what you'll remember. Now, if you have to, you'll say, ma'am, maximum confusion na yahan hua. This epidemic versus endemic. Koi test bata do, which I can use for the differentiation of these. Neal-Moser's reaction or Tunica reaction. Simple. Tunica. Tunica kahan hota hai? Tunica is in the testes. Is this some scrotal inflammation wala test? Yes. You are going to use a guinea pig, and if you inject this organism in the guinea pig is going to show you a Tunica reaction, scrotal inflammation. So where do you get a Tunica reaction? Tunica मैम अब किसमें होगा? Provaski के साथ होगा या टाइफी के साथ होगा? खत्म. Typhi is going to show you a Tunica reaction positive. So Typhi को अगर Tufi याद कर लो, if you learn Typhi as Tufi, you can easily learn that Tunica reaction is going to be positive in Typhi. Typhi मतलब endemic. Endemic typhus में positive होगा is what you need to know. It is not going to be positive in an epidemic. So remember, Tunica typhi में होता है. Tunica reaction in typhi, this is known as the Neal-Moser reaction. Out of these, I've already told you the clinical features of rash and everything, but one of them comes as a different clinical history, and that is going to be a scrub typhus. So how have I learned? Pehle to jaldi se learn. Whenever they will write scrub for you, you will take all the rhyming words. You will say shrubs, and you will say sugars. Scrub, shrub, sugars. Then you will say mite, mice. It sounds like such a tongue twister. Scrub, shrub, sugars, mite, mice. Okay, so we got that. So what do I mean by scrub? I have understood this is going to be the scrub typhus. So this is happening as a zoonotic tetrad. You need 4 things. Only then this disease will occur. Tetrad, zoonotic. Zoonotic matlab kuch animals hai. Tetrad matlab 4 cheezein. So what is it? Firstly, I need shrubs. Shrubs means that there is need of vegetations for this cycle to work. Next, we have sugars. What are sugars? Sugars are the larva of mite. Mite का जो बच्चा होता है, mite का जो larva होता है, that is known as sugars. That is why this is, I told you ना, PSM, scrub typhus is by mite. तो mite का बच्चा, that is larva is needed. Okay. What else is needed? Ma'am, mice are also needed in this tetrad. So basically, rats and squirrels are needed to carry the infection. And fourth, so can you put yourself, because it's a zoonotic disease, it will come to you. So who does zoonotic tetrad? Can I call it a sugar bone disease? Yes. So what is it? Scrub, shrub, sugar, mite, mice. Every time I have to take a stop while saying this tongue twister. Scrub for scrub typhus, shrub for I need vegetation, sugars are mite ka bachcha, that is mite ka larva, and mice is also needed. Finally, I come down to humans, so my zoonotic tetrad is complete. How the history comes is that this, whatever this sugars or this larva, whatever happens, this is gonna result in what? It is gonna result in an eschar. In which part of your body? In some moist area of your body. It could be an axilla, it could be the groin, some area where you will miss out, some fold of your body which is moist, which you will miss out. And that is the history. That there was an eschar which was there in the axilla or the groin which was not seen 5 days earlier or was missed by the clinician, was seen later, and they will give some forest visit of say Uttarakhand or somewhere, and they will give you something to do with this mnemonic of scrub, shrub, sugar, mite, mice. You know you're talking about a scrub typhus. Sorted with that. Now, are these any of these? Ye pura family, are they going to be gram staining? No ma'am, we are doing non-gram staining organisms. So किससे stain करोगे? कुछ ऐसा डॉन वाला नाम I'm gonna say. Gimsa, Jimenez, Castaneda, Machiavello. ऐसा macho नाम. 4 of these I got for you. Gimsa, Jimenez, Castaneda, Machiavello. So Machiavello basically macho name. So इतना बड़ा family था तो macho name of course. And the problem is that Rickettsia also, I'm not telling you any culture media like blood agar, chocolate agar. For chlamydia also, I did not tell you any culture media because Rickettsia and chlamydia are not grown on any of these. Rickettsia and chlamydia are obligate intracellular organisms. They will not be grown on any of these culture media. They will require cells. So either you will need some cell line for their growth or you will need some hen's egg for its growth. They will never ever be grown on any culture solid, blood agar, chocolate agar. Ho gaya. Before I end, I want to do a cross reaction. For the diagnosis of all of these, there is something called a Weil-Felix test. What is a Weil-Felix test? It is a heterophile agglutination test. Heterophile agglutination test matlab, I want to find out the antibodies of Rickettsia. From whom? From a human being. I want to find out, does a human being have antibodies of Rickettsia? That is my question mark. But antigen kya use kar rahe? Antigen I'm using OX2, OX19 and OXK. These are antigens of Proteus. These are antigens of Proteus. So, ma'am, you are doing a cross connection. For finding Rickettsia antibodies in human beings, You are using the antigens of Proteus, yes, because they cross-react, and that is the concept of Weil-Felix test. So now you don't have to learn anything. बस ये याद रख लो. All of them will show OX2, OX19, whether it is epidemic typhus, endemic typhus, any kind of Rocky Mountain spotted fever. सब में OX2, OX19 के साथ positivity. The only one that is showing OX-K positivity, and now your mnemonic is completed. The scrub typhus is going to be the only one that will show you positivity with OX-K. Rest all of them are OX2 and OX19. And that Sota Jagta wala, remember? The one that slept off and then woke up, the recrudescent, Brill's insert, that is going to show you none of these. That is the sleeping and waking, to wo apna hi busy hai. Not going to show you any of these. I end up telling you such useless stories at times, but there is possibly no other way to learn bacteriology. So yes, that is not that apna sota khata pita, that is apna separate. That is not going to get involved in this test. All the others are going to show you OX2 and OX19, and scrub typhus is going to show you K for OX-K. Finally, the treatment is going to be doxycycline or tetracycline, and one part of your rickettsia is over. But do you know, now I'm going to tell you an exception. There is something called Coxiella. You know, Coxiella is also a member of the rickettsia family, but this is not going to have any vector, any Weil-Felix test, any rash. Totally alag. Totally alag. Matlab, ma'am, no vector, so good news for us. I don't have to learn any tick, mite, louse, nothing. No vector. And when no vector will bite, if a tick will not bite me, so rash bhi nahi hoga. No vector, no rash. So how does it happen? So remember, the name of the organism is Coxiella burnetii. It can happen by inhalation. It can also happen by other routes like ingestion, inoculation, but inhalation is very, very important. So always note that this also is another eye called an intracellular parasite. Rickettsia ki tarah, all of them are growing in cells. And have I already told you what does it cause? Coxiella causes Q fever. Q because earlier it was like query fever. They did not know which organism causes it. Now they know it, but still it's known as Q fever. And this donut is also looking like a Q, because I have always told you in pathology that Q fever shows you what under the microscope? It shows you the donut granuloma. What is the other name for donut granuloma? It is a fibrin ring granuloma. It shows you the presence of fibrin ring granuloma. That is what you see with Coxiella. So donut mein se line banana, that is donut granuloma. Done. Okay, so now we come down to how are you going to do the treatment. Again, like all rickettsia, you will be using tetracycline. So that is the same treatment that you're having over here. Now, although we are still in rickettsia family, I'm going to have another set of members known as the Bartonella. That's going to end this family. The Bartonella family is also arthropod bone, so I will be telling you that, but there are three Bartonellas called Bartonella bacilliformis, Quintana, as well as Hensley. So please note, Bartonella bacilliformis, from today onwards, this is how you will write bacilliformis, because what does it cause? OR for Oroya fever. It causes Oroya fever, and that is also known as carry-on disease. So OR se to yaad ho gaya, carry-on disease or Oroya fever. And what is SF gonna remind you? SF will tell you sand fly. What is the vector? The vector arthropod that you have over here is sand fly. Easy. Bacilliformis, Oroya carry-on disease, and sand fly. What is Bartonella quintana? Quint means 5. Quint means 5. So it causes the 5-day fever or the trench fever. It causes the 5-day fever or the trench fever. Ma'am, what is the vector? 5 alpha. So I'll give you options. Louse hai, ya mite hai, ya tick hai. So you say mite mein 4 alphabets. M-I-T-E. Nahi. Tick mein T-I-C-K. 4 alphabets nahi. Kuch 5 alphabets wala batao, ma'am. Because it is causing 5-day fever. So the 5 alphabets wala is louse. So the arthropod is definitely going to be louse over here. And what does it cause? It causes the 5-day trench fever. Coming to Bartonella Hensley, where everything is going to be related to hemorrhage. Firstly, if a cat scratches you, of course, there's going to be hemorrhage. So this causes what? It causes cat scratch disease. Next, what else? It is going to cause everything to do with hemorrhage, ma'am. So it is going to cause bacillary angiomatosis, lots of blood vessels. Bacillary angiomatosis. In the liver and the spleen, it causes pileosis. Pileosis is also nothing but sinusoidal dilation. The sinusoids are getting dilated. So, matlab blood vessel is only getting dilated. So, ultimately, if a cat will scratch, there will be bleeding. If there is angiomatosis, so bleeding yaad aa gaya. Pileosis is also blood vessels getting dilated. So, all hemorrhage things are Bartonella Hensley. And what have I taught you in terms of a granuloma? What granuloma is seen with cat scratch disease in pathology? Please integrate. Cat scratch disease shows you the famous stellate star-shaped wala granuloma, isn't it? Which other disease shows you the stellate granuloma? It is lymphogranuloma venereum. And that wraps up all of your information over here. I hope everyone's okay with this. Rickettsia, chlamydia done. We were doing an MRCS mnemonic. MRCS mnemonic basically tells me that I have done with the gram-poor organisms. Spirochete ho gaya, chlamydia ho gaya, rickettsia ho gaya. The last one is mycoplasma. Mycoplasma ka mnemonic hai mycoplasma. Best one of these. Mycoplasma or the Eaton's agent is your mnemonic. So let me start. What is it about mycoplasma? Ma'am, this is the most common cause of two things. The most common cause of walking pneumonia. Walking pneumonia means patient is going to walk into your OPD himself. He's not bedridden or in the ICU. Walking pneumonia and typical to nahi hai. It is the most common cause of community-acquired pneumonia under the atypical category. See, if they would have asked you what is the most common cause of community-acquired pneumonia, I would have thought of bacteria Streptococcus pneumoniae. But if I ask you the most common cause of atypical community-acquired pneumonia, that is going to be mycoplasma or Eaton's agent. Now, this shows you a test called cold agglutination test. For cold agglutination test is seen. So we've done so many agglutination tests. I will come to a summary in the end. But why is this always said that, some people say bacteria hai bhi. Is it a bacteria? Why are they doubting it? Because number one, it's cell wall deficient. C se matlab it doesn't have a cell wall. And the cell membrane that has sterols. Dekho, bacteria don't have sterols. But this is a weird one. Bacteria have cell walls, this doesn't. Bacteria doesn't have sterols, so this has. So isme sab alag ho raha hai. That is why they're saying, is it even a bacteria? Big query on that. Cell wall is not there and cell membrane has sterols. What is PLA? PLA will remind you of PPLO. You know the other name of this organism is PPLO, pleuropneumonia-like organism. And that is why the agar that I'm using is the pleuropneumonia-like agar, on which it shows you the fried egg colonies. The PPLO agar is going to show you fried egg. But you know kya problem hai? Fried egg to ma'am samajh aa gaya. These colonies are very tiny. Why? Because सबसे पहले this organism is only SMA, mycoplasma का SMA. This organism is only very tiny. Colonies are also very छोटा. तो ma'am, how will I make those colonies prominent? I am going to add a stain called the Dine stain. So can you see in the next picture, suddenly the colonies have become a little more visible. I have added, so remember Dine stain is not what you put on a slide as a microscope wala. Dynes is what, this is a culture plate. If this is a culture plate and on this some fried egg is there, ma'am, nahi dikh raha. I'll say, okay, put a few drops of Dynes and then look at it, you will be able to see it. So that is the stain. I hope you all have not confused it with the Dynes phenomena. Dynes phenomena was seen in Proteus, isn't it? Coming back, this is mycoplasma. Quickly, most common cause of walking and atypical community-acquired pneumonia. For cold agglutination test, cell wall nahi hai, cell membrane has sterols. PLA for PPLO agar, fried egg colonies, very chota colonies, very chota organism. So I will have to use a stain called the Dine stain. Mycoplasma is pretty much done. Drug of choice bata do, that is going to be the macrolides. And I had promised you that I'm going to give you a summary of all the agglutination tests. So cold agglutination test, standard agglutination test, microscopic agglutination test. Mnemonic kya bana? There is a cat who sat on the mat. There is a cat who sat on the mat. So cold, standard and microscopic. Cat is known as Billy. So we learn it as my Billy. What is my Billy? The first one is going to be my for mycoplasma. The second one is going to be Brucella. And the third one is going to be the Leptospira. So let's get going. Cold agglutination test is seen in mycoplasma. Standard is seen in Brucella and microscopic is seen in Leptospira. Simple. Cat sat on a mat, my Billy, and these are the agglutination tests done. This is a NEET PG 2023, March 5th ko jo hua tha. That exam had the same question on microscopic agglutination test for Leptospira. Okay, so mycoplasma is done. The last set of organisms that we start with are the miscellaneous organisms, and those are going to be the bacters. Campylobacter and Helicobacter, right? They are said to be COPS. COPS matlab number one, they are catalase, theek hai, catalase positive. Number two, they are also oxidase positive. So P is going to be for positive. They are catalase oxidase positive, and they are also going to be spiral organisms, comma shape, S shape, ऐसा shape होगा. They are catalase oxidase positive and spiral shaped organism. That is what you have for bacters. Campylobacter also, Helicobacter also. Campylobacter के लिए तो best है that you learn this story, because this is going to give you a lot of information. I'll quickly tell you the story. Firstly, Campylobacter jejuni is the organism. Jejuni will tell you it grows in June ka mahina. What is the temperature in June ka mahina? 42 degrees centigrade. So Campylobacter jejuni is going to grow at 42 degrees centigrade. Jejuni tells me that. Bacter tells me they are catalase oxidase positive spiral rods. I know that. What is this Campylo going to tell you? So whenever you go camping, what are you worried about? क्या tension होता है camping में? So you will say, ma'am, camping में what will I eat? Okay, how does this happen? This is basically because of ingestion of contaminated poultry. So if you end up eating some bad things, na, you will have that problem. So सबसे पहले I'm writing down the same mnemonic over here. The mode of transmission is going to be the ingestion of what? The ingestion of some kind of contaminated poultry. So because you were tensed about food. What are you tensed more about? After food, I'm tensed about water. Water, where will I drink water from? Where will I go to the washroom from? Water and loo, watery loo होता है इसके अंदर patients are gonna have watery diarrhea. ठीक है, they are gonna have watery diarrhea anyway. Like our mothers say, जून के महीने में 42 degree centigrade, please don't go under the sun, have a lot of water because loo लग जाएगी. That is what your mother say, loo, watery loo. So remember, watery diarrhea is what these patients are gonna have. What else? Okay, now camping कर रहे हो. Camping का usually packages देखोगे ना, 1 week का package होता है. तो incubation period is gonna be 1 week. The incubation period is gonna be 7 days because camping का package 1 week का होता है. Further, if you see, camping में क्या है? You will be looking at the beautiful stars above you, isn't it? So yes, what is it regarding the stars that you will see? You will see nice effused droplet-like colonies. You're gonna see colonies which are gonna look like beautiful chota stars. They are gonna look like effused droplet-like colonies on which agar? Campylobacter agar. You're gonna see it on the Campylobacter agar. Also, there are other media like the Sciuromedia and there's something known as the Budsler media. Right? There's something known as the Budsler, Sciro or Campylobacter. So Campylobacter, you will be seeing the effused droplet-like colonies. And itna zyada worry karoge, if you will worry so much, you will end up going mad. Or CNS problem ho jayega. These patients can be associated with Guillain-Barré syndrome. Yes, there has been association with GBS also. Finally, I told you they are spiral. I can see that. I can see that they are comma shape, S shape, as a spiral. This is also known as gull wing shape. They can be comma, they can be S, they can be gull wing. And now I'll tell you something more for the stars. Which kind of motility do they have? They have the shooting star or the darting motility. They have the shooting star or darting motility, which was also shown by what? Which was also shown by Vibrio cholera. Remember, Vibrio भी comma shape था, वो भी shooting star था, ये भी comma shape है, ये भी shooting star है, correlation हो रहा है. Okay, quick revision on mnemonic. Campylobacter jejuni. Jejuni means June का महीना, 42 degree पे grow करेगा. Bacter means catalase oxidase positive spiral rod. Campy means the media is Campylobacter on which I'll see these chota stars, effused droplet colonies. Okay, the camping duration is usually 1 week. What all I worry about? Food. This is because of ingestion of contaminated poultry. Watery loo, watery diarrhea happens. Shooting star or darting motility is seen. And itna worry karoge to brain problem ho jayega. That is going to be Guillain-Barré syndrome. That is the story of Campylobacter, because now I can go on to this organism. What is this? I've directly shown you the picture. This is Helicobacter pylori. So yes, is it also catalase oxidase positive spiral rod? Yes, but there's one more enzyme which is positive over here, which I've always told you. It is also urease positive. What are the diseases that Helicobacter pylori causes, ma'am? 4 diseases it will cause. Charo stomach ka problem hai. What is it? Number 1, it causes gastritis. After gastritis, ulcers will form, to peptic ulcer disease. And 2, cancers will form. Adenocarcinoma of the stomach and maltoma of the stomach. So, sab stomach wala problems hai. But they ask you, how does it cause cancers of the stomach? It has something called CAG-A and VAC-A. What are the 2 things that God has given it for causing cancers? CAG-A and VAC-A. The last question is that they ask you, what are the different stains? So, we all know the famous Warden Starry Silver stain that you use over here, and also sometimes you can use a rapid wala stain called the modified Giemsa stain. Identify ho gaya? If not, I'll quickly revise. Warden Starry Silver stain has given it a little bit of a silver, na, to silver matlab black color. Warden Starry, let's repeat it. Warden Starry Silver stain. Ma'am, wherever the word silver will come, you will get a black color. So Warden Starry Silver stain will give it a black color. Modified Giemsa ke saath you can see this bluish color that has come. With that, the bacteria also come to an end. But before I finish off your bacteriology rapid revision, I want to quickly make you revise 3 tables of all the toxins. Tell me guys, which are the toxins? Quick revision. Which are the toxins which are gonna act by decreasing the protein synthesis? Rapid revision ka bhi ultra rapid in 2 seconds. Decreasing DPS, decreasing protein synthesis, DPS. So we have diphtheria toxin, D for diphtheria toxin, PS ko jodo to it was the Pseudomonas. Pseudomonas का कौन सा टॉक्सिन? एक्सोटॉक्सिन A होता है in Pseudomonas, that is what is there. And S and S is going to be Shiga toxin. Of course, ma'am, Shiga is going to be in Shigella. And then there is a Shiga-like toxin. Then there is a Shiga-like toxin that we have, and that is also known as the verocytotoxin. Okay, all of these are going to act by DPS, decreasing protein synthesis. But how? These two, the first two and the last two, split kar do. The first two are going to be basically causing ADP phosphorylation of EF2. That is how they cause ADP phosphorylation of EF2. That is how they decrease the protein synthesis. Whereas the Shiga ones, they are going to inhibit 60S ribosomes. They are going to inhibit 60S ribosomes, and that is how they will decrease the protein synthesis. So see, all of them decrease protein synthesis, but how is it different? Diphtheria and Pseudomonas are going to cause ADP phosphorylation of EF2, whereas Shiga ones are going to cause reduction in the 60S ribosomes. This is one set done. The second revision is which are the ones that act by cyclic AMP and GMP. तो cyclic AMP में हमने क्या निमोनिक बताया था कि M को change कर दो, it becomes CAEP, right? Twist कर दो. So number one, cholera is going to act by cyclic AMP, anthrax is going to act by cyclic AMP. We have enterotoxigenic E. coli ka labile toxin which acts by cyclic AMP, and pertussis, Bordetella pertussis acts by cyclic AMP. Then what acts by cyclic GMP? Ma'am, iska labile toxin wahan tha, to enterotoxigenic E. coli ka stable toxin is going to come up over here. How do I learn it? There's a very famous mnemonic that labile in the air and stable on the ground. Labile in the air and stable on the ground. So labile wala was cyclic AMP and stable wala is going to be cyclic ground, GMP. Further, we also have two things which I'm going to add. There is something called Bacillus cereus. Bacillus cereus causes ek to vomiting wala food poisoning. That is going to be by cyclic GMP. That is going to be by cyclic GMP versus Bacillus cereus also causes a diarrhea wala food poisoning. दो type का food poisoning, and diarrhea wala is going to be by cyclic AMP. Do you guys remember this mnemonic? How did we learn it? That anal canal के through diarrhea होगा, तो cyclic AMP will be involved in diarrhea. And गले से vomiting होगा, तो emesis वाला is going to be cyclic GMP. So repeating, गले से vomiting होगा, तो emesis वाला is GMP, and anal canal से diarrhea होगा, so diarrhea wala is cyclic AMP. This is done. Two more toxins which you've read earlier. Have you read where is toxic shock syndrome? Where have you read Staphylococcus, ma'am? Staphylococcus aureus causes toxic shock syndrome. What is the other name for the toxic shock syndrome toxin? Enterotoxin kaun sa wala? F. F will remind you of fatal disease in female. Why in female? Because toxic shock syndrome is associated with tampons. So remember, toxic shock syndrome is caused by enterotoxin F, fatal female disease. Finally, what are the toxins of tetanus? Tetanus has tetanolysin, tetanospasmin. They ask you which of them causes tetanus. It is tetanospasmin that causes tetanus. This is the one toxin that is important, right? Okay, that was a very, very rapid review of your toxins because I wanted to put it up in one place in the end, and that marks the end of your bacteriology RR. What I've also done for you guys in the notes is I've given you a final list of all the culture media, because if you've gone through this RR of 5 hours or so, after that you don't need to listen to it again and again. Just keep revising that list over and over again because your mnemonics are already with you.